Great. Um, Good afternoon, everyone. My name is Mehta, and I'm a rising senior at UMass Amherst. Today, I'm excited to share our work on using machine learning to predict chromatin accessibility regions and non-coding variant impact in human eye tissues. This work was done at the Segre Lab at Mass Eye. To start with a bit of background about open chromatin regions, or OCRs, these are segments of the genome where DNA is loosely packed, making them accessible to regulatory proteins such as transcription factors. This accessibility is essential for gene regulation. OCRs vary across cell types and are identified using techniques like ATAC-seq, a schematic of which is shown on the right. These regions contain promoters, enhancers, and transcription factor binding motifs. Importantly, OCRs also harbor non-coding genetic variants that can influence disease risk, highlighting their critical role in understanding gene regulation and disease mechanisms. One disease we focus on is POAG, primary open-angle glaucoma, the leading cause of irreversible blindness, which affects over 75 million people worldwide. POAG is characterized by gradual retinal ganglion cell death and progressive optic nerve damage, and is often associated with elevated intraocular pressure, though 40% of patients also have normal pressure. Current treatments for POAG are limited and not effective for everyone, which highlights the urgent need for new approaches such as neuroprotective drugs. Interestingly, POAG is highly heritable, suggesting a strong genetic component we can explore through chromatin accessibility. So why use machine learning? Genomic data is extremely high dimensional and complex. Traditional methods like linear regression easily can't handle the nonlinear patterns often seen in regulatory DNA. So our study aims to address the following questions. One, can we train a model to classify open versus closed chromatin in a set of 4 eye tissues? Two, does this model capture tissue-specific patterns, especially within the broader HuBMAP dataset? And three, can the model help predict the regulatory impact of non-coding variants linked to POAG and intraocular pressure? For this project, we use bulk ATAC-seq data from 4 human eye tissues: the ciliary body, trabecular meshwork, optic nerve head, and macular region. These 4 tissues are key tissues for the pathogenicity of glaucoma. Ciliary body and trabecular meshwork are aqueous humor outflow pathways that regulate intraocular pressure, and drainage in these pathways can lead to elevated IOP and glaucoma. The retina and macula harbor retinal ganglion cells that degenerate in glaucoma. These RGCs exit the eye to the brain via the optic nerve head. Our data was provided by the Cherry Lab at Seattle Children's and was carefully quality controlled. We also applied a custom in-house denoising pipeline to enhance the signal-to-noise ratio for our peaks, along with removing problematic regions such as repeats and gaps, which gives us a clean dataset for model training. On the right, we can see a distribution of peak lengths from within the denoised ATAC-seq peaks. Ciliary body, macula, and optic nerve head have a median peak length of around 1550 base pairs, whereas trabecular meshwork has a higher median peak length of about 2250 base pairs. For our modeling, we use GKM-SVM, a method that finds motif-like patterns in DNA sequences. It works by breaking the sequences into small 11 base windows called 11-mers, and then converts them into gapped k-mers to capture flexible patterns in the genome. These are compared using a kernel function to measure similarity between sequences. We trained the model using a set of positive regions, which is open chromatin peaks, and an equal number of negative control regions that come from non-open chromatin to serve as a background. To validate our approach, we first trained a proof of concept model using single-cell ATAC-seq data from the human retina. We focused on 8 distinct adult retinal cell types, including both mature and progenitor cells. After pre-processing the data, such as standardizing peak lengths to 301 base pairs, we trained our support vector machine model. The results were strong. We achieved a high ROC-AUC between 0.95 and 0.97, indicating excellent performance on single-cell data. To apply our model to bulk data, we applied the following pre-processing workflow. We standardized peaks, removed universally shared peaks, and used background regions, which is our negative set, which were length and GC matched to a set of positive ataxic peaks for each tissue. However, when we applied the same approach of utilizing 301 base pair regions, bulk ataxic performance dropped significantly, with ROC AUC values around 0.67 to 0.73. This highlights the added complexity and heterogeneity present within bulk tissue, which can obscure cell type specific regulatory signals. To deal with this, we updated our pre-processing approach to preserve all peaks up to a maximum of 13 kilobase pairs. The updated classifier was reasonably accurate, displaying a ROC AUC of 0.81 through 0.90, and a similar ROC AUC for precision recall. The model was also found to be well calibrated, providing confidence in its predictions across eye tissues. Next, we began looking at tissue specificity for tissue that's very different from eye tissue. We identified a pancreas dataset from ENCODE, which we used along with our outgroup set from our bulk tissue peaks to identify differences in SVM prediction scores. We found significant differences across tissues for ciliary body, macula, and optic nerve head. This was validated via a Wilcoxon-Ransom test with one-for-one correction, suggesting our model captures biologically meaningful tissue-specific patterns. And we are also gearing up to test these on single-cell ataxic data from HuBMAP to identify cell types with similar open chromatin region patterns. For our final research question, we implemented Delta SVM, a method for scoring the impact of non-coding variants on chromatin accessibility. The goal is to predict whether a variant might disrupt gene regulation by altering the openness of the surrounding DNA. We began by generating a vocabulary of scores for every non-redundant 11 mer using a model trained on our open chromatin regions. These K-mer scores represent how much each sequence contributes to making DNA accessible. For each variant, we identified or extracted a 301 base pair window centered on the variant and created both the reference and alternate sequences. We used our trained vocabulary to rank and aggregate the results of the effect of a variant. The delta SVM score is calculated as the difference between the alternate and the reference score. A negative score for delta SVM suggests that the variant reduces accessibility, possibly disrupting gene regulation. Variants with strongly negative scores are prioritized as potentially functional. Next, we applied our model to score 747 genetic variants associated with glaucoma-related traits. These variants came from several large GWAS studies, as shown here. We focused our analysis on the most negative scores from models trained on two tissues, macular and optic nerve head. These are relevant to the optic nerve and are likely involved in neuroprotective responses independent of intraocular pressure. One variant of note is R742243, which co-localizes with an expression QTL for the gene TRP2 in fibroblasts, as shown by Hamel et al. In 2024 and Han et al. In 2023. TRP2 is homologous to serine threonine kinases and is known to induce apoptosis, mainly in cells of hematopoietic origin. This suggests that they may influence signaling pathways involved in cell death or immune response, potentially linking genetic regulation to disease progression in glaucoma. We're also currently working on comparing the delta SVM scores to a null distribution to assess which variants are likely to have a significant regulatory effect. To summarize, we trained machine learning models like GKM-SVM, and we found that they were effective to classify sequence patterns based on levels of chromatin accessibility. Variant impact can be quantified using delta SVM scores to gain insight into putative causal regulatory mechanisms. And our trained models reflect organ-specific regulatory signals, which enables variant impact prediction within a relevant and reasonable biological context. Moving forward, we plan to test our models on HuBMAP single-cell data to identify cell type-specific signals, also quantify variant impact scores, and compare delta SVM with other model interpretability tools, and integrate deep learning models such as ChromeBPNet Alpha Genome to enhance performance. I'd like to thank Dr. Rinaldo Catapreta, my mentor for this project, and our PI, Dr. Ayelet Segre, and the bioinformatics team, and all members of the Segre lab. thanks to our collaborators at Seattle Children's Hospital and Tim Cherry's lab. And special thanks to the NIH and HuBMAP consortium for providing me this amazing opportunity, to the University of Florida for their smooth organization. Thank you so much for your time, everybody. I'm happy to take questions.
Very interesting work. Had you worked in eye health or eye biology before, or was this your first time learning about it?
So my background is in computer science. The whole domain of bioinformatics was kind of new for me, and not to mention eye tissues. But it's a very interesting challenge, and I really enjoyed working with it.
What was your most interesting finding?
I guess the most interesting finding was the variant impact scores that we talked about, because those, a lot of models currently exist, which provide some kind of training and which provide some kind of accuracy scores for tissues, but to actually quantify them and to get a result that is hopefully biologically meaningful and can be also used downstream in potentially clinical results. So that's the most exciting part.
Well done. Thank you.
Thank you.
What was the biggest challenge that you faced working on this project?
So there were several challenges, but I guess the foremost was to identify a good trained model. We applied a similar workflow. We applied a workflow taken from one of the papers that uses GKM-SVM on our data, and we did not find the ROC results as they had in their paper. So we had that kind of problem about trying to train the model to be more generalizable. And that's the biggest kind of thing we iterated upon.
There are no other questions. I have a quick one for you. What do you think is the most valuable thing you learned from the course that you'll take in your research journey?
I think it's a kind of a naive answer, but I did not know what RSID was, and the first module on the course was creating an RSID ID, and that was so it's these like nuances about research and about presenting yourself, which were really helpful for me to kind of gain experience with.
Definitely research will get you. There are a lot of little tidbits that you learn along the way, which is why mentors and lab work, working as a team is really important, because if you don't know it, I promise you someone else does. But great.